Previous class, I have explained the separation and identification of the proteins by using mass spectrometry. So now the today class is how we use the mass spectrometry for identification of the protein mass. So mass spectrometry is one of the tool in proteomic analysis to study the protein mass. So it is analytical technique that measures the mass bar charge ratio of the charged particle in a vacuum. Mass spectrometry can determine masses charge ratio with high accuracy. Molecules in a test sample are converted to the gaseous ion that are subsequently separated according to the mass charge ratio. In mass spectrometry, one of the main application is it separate the molecule based on their charge and mass ratio. Several types of experiments can be performed with mass spectrometry. So the basic principle involved in this technique is organic molecules are bombarded with electron converted to highly energetic positively charged ions. Molecular ions are the parent ions. Further break up into smaller ions, fragment ions or daughter ions. The formed ions are separated by the deflection in magnetic field according to their mass and charge. Finally, it reads the mass and charge of the protein in a mass spectrum. This is the simplified scheme of the mass spectrometry. In this spectrometry, first we have to put the sample. So sample undergo the ionization. Ionization means by flowing the electrons, the proteins are the component of the amino acids. Amino acids exhibit the different side chains. Based on the side chain molecules, the amino acids are ionized as a positive and the negative charge. So the protein molecule are ionized into different ions. Next, these ions are passed to the mass analyzer. This mass analyzer, it separated the ions into different based on their mass. Here you can observe the different colors of the dots. Purple color, blue color. So that means based on their charge and mass, these are ions are the separated. So the first low molecular weight ions are moved faster than the high mass ions. So finally there is a detector is connected to the technique. This detector detect based on their size and charge. So again there is a software is connected to the detector. In this software, it gives the different peaks based on their size and mass. M by Z ratio. This is called the M by Z ratio. M by Z means mass and charge. So this is the overall equipment. The cations that are formed are separated by the magnetic deflection in mass spectrum. So this is the probe. Probe means it passes the ions. Ion source sample. This is the sample holder. Here we have to place the sample. So the sample, during the sample passes through the equipment, it passes, there is electron beam. Electron beam, it passes the beam of the electrons. By using these electrons, the surface charged amino acid molecules are ionized by accepting the electrons or donating the electrons. After that, the ionized amino acids present in the sample, they are hit the accelerated plate. This accelerated plate is slit into two parts. There is a slit, entry slit and exit slit. This is totally called as a accelerated plate. So this is the ion beam. Ion beam means after exposed to the electron beam, the amino acid component of the protein are converted into the beam of ions. These ions are passed to the magnet. In their magnet, based on their mass, they are split into the different waves. Ions that are too light bend too much. If the ions which are too light, that means the ions which are charged by passing through the beam of ions, these ions, if it has a low molecular weight, that means low mass, the beam of the ions are split into the side, bend, like it forms as a bend. The ions which have the high mass, ions that are too heavy bend to little. So these ions are passed through the magnet. So after that, this is called as a flight tube. This is the TOF, okay, time of flight. So only ions of the right mass can enter the detector. The ions, after separating with a different charge, they are can enter the detector. So there is a vacuum pump is there. So there is a detector slit. This detector slit, the detector ions are passed into the detector slit, and there is a connection of the detector. Finally, detector detect the mass of the protein. So the resulting mass spectrum is a graph of the mass of each cation versus its relative abundance. The peaks are assigned an abundance as a percentage of the base peak, the most intensive peak in the spectrum. The base peak is not necessarily the same as the parent ion peak. The mass spectrum of the ethanol. This is ethanol. Ethanol is a C2H5OH is the molecular structure of the ethanol. Ethanol into split into the different peaks based in the ions and mass. So whenever the beam of the electrons are passed the ethanol into splitted into the three peaks. This is the mass peak and this is the base peak. So this is the technique used for identification of the protein in a proteomic tool.